Welcome, everyone. My name is Melissa Comcar, the Senior Director of Customer Success at Emulate and the moderator for today's webinar. I'd like to thank you all for joining our webinar on how to get started with organ-on-chip technology with our Senior Director of Product Management, Luke Dimasi. Today, you will learn about Emulate's organ-on-a-chip technology, its benefits compared to 2D cell culture, and data-driven examples of organ chips' improved physiological relevance. Before we get started, I want to go over a couple of housekeeping items. If you have any questions, I ask that you please submit them in the Q&A box, and we will answer as many as we can at the end of the webinar. The webinar will be recorded and sent via email after the webinar concludes. At this time, I would now like to hand the presentation over to Luke.
Thank you, Melissa, and hello, everyone. As Melissa mentioned, my name is Luke Dimasi. I head up the product management team here at Emulate, and I am very excited today to be giving you an introduction to organ-on-a-chip technology, and then walking you through some of the benefits of the technology and how you can apply it to extend both the functionality, complexity, relevance, and predictive power of your current in vitro cell culture models. So in terms of an agenda today, as Melissa mentioned, I'm going to do a quick overview on the current state of play of cell culture technology. So we'll touch on things like 2D cell culture, the evolution of cell culture into 3D systems. I'll then go through some of the benefits of organ chips and how you can use it to improve human relevance, and that includes an overview of the system itself. We'll talk about clinically relevant endpoints and the amount of data that you can get off of organ chips, and then how to get started in terms of dipping a toe into the organ chip world, and how you can start to look at translating what you're doing today and then evolving that onto organ chip technology. Okay, so let's take a look at the state of play right now, 2D versus 3D. So when it comes to things like disease modeling, identifying targets for drugs or therapeutics, therapeutic development as well, right now, the kind of state of play, the technology that the industry, the academic, the research industry, as well as the pharma industry rely on, are primarily in vitro technologies like cell culture, 2D cell culture, or animal studies. And these can be small animals like mice and rats, but also larger animals as well, like beagles, as well as non-human primates. And these types of models have had a long heritage in terms of looking at not only identifying new targets, but then how we drug them, and then looking at the efficacy and safety of those drugs as well. But we know that these models have problems. They typically do not translate the responses they see into human in vivo responses, and we see that as a translation gap. At Emulate, we believe that the future is delivering true human biology to the research bench using organ chip technology. So I touched on these models before, and I said, there's this translation gap, but what does that really look like in terms of drug development? And this is well known in the industry here. It takes over a decade to bring a new drug to market, from discovery all the way through to regulatory approval, at a cost of $2 billion in terms of R&D costs per drug candidate. But the issue here is this failure rate. There is a 90% failure rate of drugs once they get into the clinic. And the failure is attributed to either efficacy, they just don't work, or safety issues or toxicity issues. So only 1 in 10 drugs actually get to market, which is a 90%, basically a 90% of R&D spend that is not optimally allocated. And we believe the reason for this is the preclinical models that are used in development, in discovery development, and preclinical development here. So we see that there's an immense opportunity here to improve the models and the human relevance and translation in these preclinical side of the story. So let's talk about the evolution of in vitro cell culture. As I mentioned, it's one of the two pillars that people use in terms of looking at targets and drugging targets, that in animal studies. But in vitro cell culture has had a long heritage of improving its complexity, improving its relevance over the last few decades. Initially, cell culture started off with the propagation of mammalians' cancer cell lines. These were easy to grow in the lab. They were mammalians, so they kind of had the relevance that researchers were looking for, and they were good to look at genes and really probing individual pathways and looking at it within a cellular context. Primary cells were also another biological input or technology that the research community used here. These were a little bit more relevant. They maintained their phenotype longer compared to cancer cell lines, but they were difficult to propagate in vitro, and even though they're still used today, there's lot variation because you have to keep going back to extracting these from the primary organism for culture. I would say a step change in vitro cell culture came with the introduction of 3D modalities such as spheroids, specifically tumor spheroids. Here, the industry was starting to model more sophisticated microenvironments. We're starting to see cells as they normally would be in the body, and we're starting to see effects such as gradients and even hypoxic cores, which play a role in terms of influencing that biology. So we're starting to get a little bit more relevant as we continue. Another step change is the introduction of iPSC technology. This essentially overcame many of the issues with primary monoculture. Here you have an inexhaustible supply of any cell type, and the benefit of this is you can get those somatic cells from a patient. You can reprogram them to an iPSC state and then force differentiate them to any terminal tissue. So again, an inexhaustible and scalable way of looking at generating a lot of biological material in a consistent way. Organoids, another step change here. So in here we're starting to talk about a heterogeneous and complex set of cell-to-cell interactions that are starting to take place here. Organoids can be derived from primary biopsies and then propagated in vitro, and they contain a stem cell population that's able to continually replenish that heterogeneity, that cell source. They can also be derived from iPSCs. But they're another step change here, because not only are we starting to model microenvironment, but we're starting to add more complexity, more cell-to-cell interaction here. We see organ chips as the next logical step change here in driving in vitro cell culture to greater human relevance. You can take that diverse, heterogeneous cell population that's present in organoids or any cell input, and then you can start to look at applying microphysiological forces to drive those cells to a better state of functionality, adding more complexity, and then improving the predictive power of the response of the model. And I'm going to show you how that can be done over the next few slides. So again, just to take a look at those preclinical models that are currently the bread and butter of the industry. Animal models, again, a long heritage, really good in terms of looking at systemic drug effects assessments, toxicity, efficacy. But we know that the non-human relevance there and the species difference is one of the primary reasons for translation failure. In fact, almost 80% of researchers out there have wavering confidence in the translation abilities of animal models. Static 2D cell culture, again, another in vitro technology, preclinical technology, in vitro technique, inexpensive. Cells can be propagated. You can look at high throughput screening, so you can look at querying a lot of different pathways at the same time, or even drugging with a lot of different drug candidates at the same time. But we know the downfalls are limited cellular complexity. Often cancer cell lines are used. They don't have that in vivo phenotype or maintain it. And there's no mechanical forces here. So you're dealing with kind of immature models. Organoids, as I mentioned, improve that functionality, that complexity over 2D. They have that rich and diverse cell population, so you can see that crosstalk between cells. But there are the downfalls. They're highly variable. They're expensive, hard to propagate in vitro. They don't often have endothelium attached. So although you have cells from the organ, you're not really modeling a functional organ unit. And there's no mechanical forces there. So again, you're not driving these cells to be differentiating and maturing in the way they would in the body. So we believe that organ chip technology is a way to extend this biology and open up a new window in human biology. So let's talk about the fundamentals of organ chips. So first, there's tissue-to-tissue interface. Organ chips allow you to have a separate epithelial or parenchymal chamber, as well as a separate endothelial or vascular chamber. We have workflows that enable consistent and controlled seeding of cells across those two chambers. And we have a porous membrane that separates them. So it enables the crosstalk between cells, between chambers, as well as you can see cell migration occurring, either tumor cells, for example, moving from the parenchymal space into the endothelium or vasculature, or even immune cells moving from the vasculature and recruiting into the epithelium. It's a dynamic environment. Independent unidirectional fluid flow drives the maturation, and it improves the functionality of the biology. You can see the effluent coming off of the chips over time, so you can sample that effluent. You can see that the change in biology over time. And mechanical forces stretch that biology so you can get that maturation and functionality, and we can see that. And I'll show you some examples later. Also, the organ chips are flexible. You can add complexity as needed. We have workflows for applying and circulating immune cells. As I mentioned, you can look at recruitment of immune cells from vasculature into the epithelial or the parenchymal space where they have their effective function is exhibited. You can add stromal cells, you can add microbes, even hypoxic workflows. And a lot of our research partners have applied these workflows to extend their biology. Oops. Yeah. Okay, so let's start with the core of our technology, which is the S1 stretchable chip. So I'm going to take you through a little bit of a cross-section of the chip. As you can see, there are two chambers, an upper epithelial parenchymal chamber and a lower vascular or endothelial chamber. These chambers are separated by a porous and flexible membrane. This enables crosstalk, signaling between the chambers and cells to move back and forth. You can seed each independently. And then our system applies vacuum to the chip, so we're able to stretch that biology. And this is important for models such as intestine, which has a peristalsis, or lung. You're able to maintain each of these cells in their preferred media environments, and you're able to sample this effluent over time to see changes in that biology, either due to a gene change or a drug change. So that's the organ chip. Let me show you how it interfaces with the greater human emulation system offered by Emulate. So as I mentioned, we have our stretchable chip here. It is transparent, so you can image through it. It is flexible, so it can stretch and apply those forces to certain organ models. Two independent chambers that are over each other. The chip sits in a chip cradle, and then this connects to what we call our pod. And the pod has the reservoirs that drives that media, those fluids through the chip. 12 pods can go into our Zoe culture module, and this module here then basically sustains that biology. The manifolds come down. You can see it's a seamless organ chip interface. We don't have things like pumps or tubing, so it's a really seamless, easy to use system that makes organ chip accessible for people. The culture module itself is supplied power as well as gas control and vacuum with our orb. And the orb also has the ability to provide warnings and notifications. And we have a suite of software that helps drive the workflow, control the Zoe, as well as we're building out a suite of data analysis tools to make the interpretation of the data you get off the chips a little easier. So that's the system. Let's talk about how you can now use it to improve human relevance of your biological models. So there's 6 things I'm going to touch on. I'm going to talk about how it's an open platform. It's compatible with a wide variety of cell sources. I'm going to talk about the importance of a tissue-specific endothelial interface and co-culture, as well as flow. We'll talk about that you are able to tune mechanical forces or apply mechanical forces and why that's important. I'll show you what that looks like in terms of improvements to gene expression and what that does to the predictive power of the model. So starting with the open platform. So at Emulate, there's a number of ways to get started with organ chip technology. We have already pre-packaged and validated models that are available to purchase from Emulate. There's 5 in total. These can be purchased through what's called a bio kit, which has all the consumables and the pre-qualified biology. So we have a brain chip, which models a neurovascular unit. We have 2 intestine chips, duodenum for the upper intestine and colon for the lower intestine. We have a proximal tubule kidney chip and a liver chip. So as mentioned, these are pre-packaged, ready-to-use chips to help customers get up and running in a quick way with organ-on-a-chip technology. But the system itself is extremely flexible. And in fact, our users have applied biology to all sorts of biology to chips. In fact, there are over 100 publications out there referencing over 70 organ models in existence. So the system is extremely flexible in terms of accepting different types of biological inputs and modeling different types of systems. In terms of cell sources, the chip is compatible with a wide variety of cell sources. So, we touched on a few of them before: cell lines, primary cells, induced pluripotent stem cells, even 3-dimensional models such as spheroids and organoids. We have workflows for all of these cell types that can be applied to chip. For spheroids and organoids, we're able to break apart and fragment those sort of complex 3-dimensional models, and we have workflows to see them in a very consistent way. They offer that rich and heterogeneous cell population to really start building sophisticated models. As mentioned, increased cellular complexity with co-culture with the relevant endothelium is also possible on chip. One of the examples of our more sophisticated chips is our brain chip, and this is one we launched last year. This chip has 6 cell types that are part of the biology. There are 2 iPS-derived neural cell types, neurons. There are primary astrocytes, primary pericytes. We have a microglial cell line in there. We have an iPSC-derived brain endothelial-like cell line as well. And this is what we pull together to emulate the neurovascular unit. And we use this for looking to model neuroinflammation. So again, the chip can take all sorts of cell types and biological inputs, and we can guide customers on how to adapt their current models to working on chip. So tissue-specific endothelium, and why is this important? And I'll give you a couple of examples here with organ models that we've developed here at Emulate. Starting off with our colon intestine chip, which we launched last year. So, when we match and the colon intestine chip is the epithelial layer is a primary biopsied organoid material, and the endothelial layer is matched colon microvascular endothelial cells. So we do this on purpose to match the cells. We think it's very important to recapitulating that complexity and that crosstalk. And you can see here on the left that when you have endothelium present, you get a nice tight barrier that is established earlier on in the model, and that barrier is also present in the human colon. It separates the epithelial, the colon itself from the vasculature. And when there's disruptions to that barrier, all sorts of disease pathologies exist, and we can model that as well. But the endothelium also improves that cell morphology. We see that not only does the barrier look better, but we see a formation and maturation of the epithelia. We have a polarized brush border and densely packed networks of elongated microvilli. So the epithelia just looks better and it starts to recapitulate a more in vivo-like state. Similarly, with our kidney chip here, we can see we were looking at when we were looking at building this, we were looking at making sure that the kidney epithelial cells were expressing the sodium phosphate cotransporter. It's an important transporter, functional transporter for kidney epithelial cells. In monoculture, you can see on the left-hand side, the expression levels are relatively low. When you add HUVECs, which is a non-organ matched endothelial cells, these are just human umbilical vascular endothelial cells, you see that we don't get any improvement in the expression of that transporter. In fact, it looks worse with the wrong matched endothelial cell. But when you match these renal epithelial cells with a renal endothelial cell, you start to see that signaling occurring between between those two cell types, and you see an improvement of the expression of that sodium phosphate transporter. Again, pointing towards a more functional epithelial biology. So as I mentioned, two examples here of how endothelial cells can improve the look and the functionality of the cells, but it also opens up new mechanisms of probing the biology. And here's an example that was published last year by Thacker and colleagues. Here they were looking to model an alveolar lung chip. Now, typically in conventional monoculture air liquid interface systems, alveolar cells aren't readily infected by SARS-CoV-2 in vitro. They saw that on chip, they were able to get robust infection of SARS-CoV-2. So these alveolar lung cells were clearly thriving, starting to express the right types of cell surface receptors for the virus. But when they started modeling this with the relevant endothelium, they were able to look at not only the infection of the epithelia in the relevant way that the SARS-CoV-2 actually infects the lung tissue, but they were able to look at the knock-on effect of that infection on the endothelial cells. So the SARS-CoV-2 virus started to infect the endothelium, and they identified a novel mechanism that was resulting in blood clot formation as a direct consequence of that endothelial cell infection. And this is something that wouldn't have been possible to model in a monoculture scenario. Another example of looking at opening up new mechanisms with relevant endothelium and flow is an application we actually launched last month here. This is colon inflammation and looking at colon immune cell vascular recruitment. So in the inflammatory process, as a tissue becomes inflamed, immune cells normally move from the vasculature into their epithelial space. It's done through a very controlled sequence of events. But what's important to drive that specificity of response, both inflammation and gut-specific response, is the flow and that shear that occurs in the bloodstream. So immune cells don't just randomly exit the bloodstream into the epithelia, otherwise they'll cause all sorts of autoimmune reactions. But this shear acts as a gate. So in the body, if the signaling isn't strong enough, the cells essentially will just continue to flow through the vasculature. And we're able to model that in our colon intestine chip with underflow. So here you can see on the right-hand side, we have flow. We have the flow in the vascular chamber at the necessary level of shear. We see those PBMCs start to roll across the endothelium. When we prime the model to become inflamed, the endothelial cells start to project their adhesion molecules that roll. The immune cells start to arrest, and then they start to migrate through that endothelium into the epithelial chamber, and we're able to actually quantify that movement. And then once they get across, we can actually see the effector function of those immune cells. They start to express and release their cytokines, and we're able to see that them kicking off that inflammatory process and how that damages the barrier of the colon. And that's what happens with diseases like IBD and other inflammatory gut diseases. So flow and endothelial cells are imperative to being able to model this in a relevant way. Now you can actually start to perturb the system, looking at therapeutics that can stop or mitigate that recruitment and activation of immune cells. So that's the importance of both the matched endothelial co-culture as well as the flow. Let's talk about now the tunable mechanical forces. So why is stretch important? So again, when we looked at developing our colon intestine chip, we could see that stretch was important to improve that differentiation, the maturation of that epithelial tissue. When you put these cells on just a sort of a static transwell system with no stretch, they take on kind of a flat morphology, which isn't what they look like in vivo. When you start to apply that flow and that stretch, you see an improvement of the epithelial cell height and the polarity of those cells. So the transporter's in the right place, and they're starting to look more in vivo-like. So we can see here that that stretch and flow are important for driving that the epithelial maturation. So the epithelium looks better, but again, stretch enables you to open up new ways of probing mechanisms here. So in this example, and this was published by Grazart last year, here we're looking at Shigella infection. And here in the publication, they were showing that in fact, intestinal flow and peristalsis are important for driving the mechanism of how Shigella infect the epithelium. So we're looking at identifying new mechanisms of infection, which then enables you to look at ways of perturbing those pathways and those mechanisms. Another example is enabling tunable models for tumor microenvironment modeling. And Strelez and colleagues published this in 2021, where they were looking at the invasion capacity of colorectal cancer on a KECO2 intestinal chip. So, and here they were showing that peristalsis motion was important for driving that invasion of those cells into the epithelia and then subsequently into the vasculature. Not only that it resulted in differences in terms of the metastasized tumor cells, and that impacted their ability to travel and then start to exit that vasculature and in distal locations to cause metastatic growth. And they demonstrated this with the intestine chip as well as a liver chip in terms of the recipient of those cells. Here they did this on a KECO2 model, and again, it's a very simple model, easily transferable from transwell to chip. But this group here is now looking at expanding on this model with primary biopsied organoids here, so becoming a little bit more in vivo relevant and looking at colorectal cancer metastases in that model. Okay, so we talked about how endothelium and flow is important for driving that functionality and looking at new mechanisms, and how stretch is also important for driving maturation, functionality, and looking at new mechanisms of biology. Here we're going to show you how how that impacts the gene expression profile of the model. And again, just looking here at our colon intestine chip, we're looking at the transcriptomic profile. This is the expression profile of genes on chip, as well as in organoids, just primary biopsied organoids maintained in suspension culture. And as you can see by looking at the transcriptomic signature distance, that when you culture these primary biopsied organoids on chip, up to day 8, you can see that they maintain a closer resemblance in terms of transcriptomic signal distance to in vivo human colon, versus organoids, where you're seeing that drift away from what the in vivo transcriptomic profile looks like. So we can see that, again, the transcriptomic profile looks more similar to what's happening in vivo. And it's an improvement over suspension organoid culture alone. We've also seen this not only in colon chip, but also in our brain chip. And we can show here that the microenvironment of the brain chip, again, enriches several biological processes versus transwell. So we see an upregulation of key genes. And if you look at the gene ontology here, the enrichment of genes are those that point towards maturation of the model. And we see this on chip versus transwell. So again, those microphysiological flow of the complexity of the model with the matched co-culture with the endothelium is important for driving this maturation, and we see this in the gene signatures. Also, when we look at sort of inflaming that brain chip, so we have a healthy chip, and then we sort of induce a pathological inflamed state. With TNF alpha, we see an upregulation of the relevant genes that are a response to that inflammatory cue. So this includes astrocyte migration, response to the chemokines, response to interferon gamma, glial cell activation. These are all the relevant gene types that are upregulated as part of the pathology we're trying to emulate here. So again, if you want to get started, Emulate has a number of transcriptomic signatures of all of our on-market chips. So if you want to see if your particular gene of interest is present in the healthy chip as well as in a pathological state, we're able to show that, and then they'll sort of give you kind of a flavor of is the model actually recapitulating the biology that I want to see. Okay. So we showed that we have better morphology, we showed that we have better functionality, we showed that we're opening up new mechanisms to probe, we're showing that it has an improvement in gene expression. But so what? At the end of the day, what we care about when we're talking about building a more complex model is, does it improve the predictive power of the model itself? And this is something that Emulate wanted to go out and prove last year. And in fact, we did that with our liver quad culture kit, a quad culture chip. So this was a study that we initiated. It was one of the largest we ever performed. It was 780 chips, liver chips. And we wanted to validate this model with 27 known hepatotoxic and their non-toxic analogs. We didn't just cherry pick these drugs. These were selected. The selection was guided by the IQ-MPS affiliate. This is a consortia group of pharmaceutical companies that are interested in standardizing and developing microphysiological systems, MPS systems. So they came up with a list of drugs that they said, if your system can accurately identify toxic and non-toxic drugs out of this list, they consider it a good system to be able to move forward for use. So we took that as our initial list, but we also want to enrich that list with drugs that we knew that slipped through both animal studies as well as the next best-in-class model, hepatic spheroids. Now, 2D hepatic cell culture as well as hepatic spheroids are being implemented for tox screening. They pick up the bad players, but some slip through these false negatives. And we've indeed seen this as part of other published works. So we took that list, we looked at the major mechanisms of drug-induced liver injury, or DILI, and we tested all these drugs in a blinded way. So they were optimized for relevant Cmax as occurs in vivo. And we profiled them in a blinded way so that the people that were actually executing on the screening didn't know what compound they were actually dosing with or analyzing until the end by an independent group. And this was published last year and is available right now on bioRxiv if you want to get into the detail of the study as well as the results. But here is a summary of the results and the impact there. So I'll draw your attention to the bottom part of this slide here. What we showed with the quad culture liver chip with 2 donors is that we were able to achieve an 87% sensitivity and 100% specificity in terms of correctly identifying toxic and non-toxic compounds. And this is matched a marked improvement over spheroids, which are only able to have a 47% sensitivity in terms of correctly identifying those things. So again, here we're showing that with improved complexity of model, that we're able to see an improvement in the predictive power of the model. And in fact, we advocate that liver chip should be a standard part of that drug development value chain in terms of looking and profiling the vital few compounds before moving into clinic for their DILI risk. So that shows you how that with improved complexity, you do get a marked improvement in the predictive power of the model. And this is proven on the liver chip, but also this can be demonstrated on other types of models as well. So let's just pivot for a moment to the types of data that you can get off of the organ chip. And we say clinically relevant endpoints. Organ chip technology enables you to profile the biology over time. You get an extremely amount of detailed depth of data from an organ chip. There are a number of classes of data you can get. As I mentioned previously, effluent collection is one of the main ones, and you can do this over time. You can sample that biology over time, see how it changes. We use effluent collection to look at the barrier function of the model. So this is important for things like the gut, as I mentioned previously. A healthy gut has a nice tight barrier between the epithelia and the vasculature. When you induce a pathological state, that barrier breaks down, that microbes and all sorts of nasties start to get through. So that's effluent collection is the way that we look at that barrier in a temporal way. But you can also see cytokine release from each chamber independently. You can look at the metabolomic profile of the biology, and we also use it to look at the injury markers for our liver chips as well. So typical markers includes transaminases, albumin production. These are the things that people look at in vivo and in clinic to gauge liver function and dysfunction. The chips are flexible and as well as transparent. So you can start to image a number of different modalities: phase contrast, confocal. You can do live stain imaging as well. We have workflows that enable that. There's workflows for electron microscopy as well as H&E staining as well. And just a couple of examples, you can see the images here are looking at the epithelial chamber top down. On the left-hand side, and you're seeing immune cells actually start to, that are stained, they have a live cell stain in them. You can see them in red starting to poke through the epithelia. But we have a workflow and an analysis technique for looking at the cross-section of the chip. That's what you see on the right-hand side there, this cross-section. We can see the amount of cells that actually attach to the endothelium. We can see them as they start to traverse that endothelium in their migration, and how well they migrate, and we can see them as they actually get to the top there. So we're able to quantify the movement of cells over time, which is quite nice. And finally, gene expression. We have workflows for extracting that biology, so you can do bulk RNA-seq. In fact, that's the major mechanism that we use here at Emulate, but we're also looking to move into single-cell RNA-seq, and there's workflows that enable that, as well as proteomic analysis. So again, rich data you can get off of the chip. This is not a simple assay with a yes/no answer. You're able to really probe the system in a number of different ways to generate a picture of what's going on with that biology. So how to get started? And as I mentioned before, Emulate offers kind of an end-to-end solution. We have the chips themselves, we have kits that with pre-validated biology to get you started, and we have a system that helps really make it easy for an in vitro cell biologist to start to pick up and get into the organ-on-a-chip world. So you can adapt your current model to organ chips quite easily. We provide the equipment, the consumables, as well as the expertise to get started. Emulate offers two instruments I showed before, the Zoe culture module. Zoe means life. It sustains the life on the chip. And the Orb hub module. The Orb powers the Zoe. It provides the gas as well as the vacuum. You can scale out. So one Orb can power 4 Zoes. So if you want to start with one Zoe, you can start to scale out as you exhaust the capacity of that Zoe. We have a basic research kit, which contains the chips, the pods, all the reagents for the workflow. And then you can provide your own cells, and we show you how to again, adapt your technology, your model onto that with our expert scientific support. That includes hardware support, protocol generation, as well as scientific training and experimental support as well. As I mentioned, we have pre-qualified organ models ready to go. This will help you get started quickly in terms of building an organ model, so you don't have to start from scratch. So we have brain, we have 2 intestinal models, we have kidney, and we have liver. And again, once you get started with and get comfortable with that workflow and establish that biology, you can then start to substitute your own cells or different cells with the cells that we provide in kit, and start to really tailor that biology to your specific scientific question. If you're interested in just getting to the data, the rich data that comes off organ-on-a-chip technology, we also have fee-for-service offerings. So we can do these on our standard organ chip offerings. We tailor the models as well to a limited degree. We can also do custom model development for in certain scenarios. But off the shelf, we have standard toxicity testing. We can look at target validation, efficacy evaluation, mechanism of action studies, rank ordering of compounds, and we can customize the data analysis to support your needs as well. We can provide raw data with all those endpoints, as I mentioned before, or we can package it up into a nice report that's easily digestible. So that's how to get started with Emulate technology. But what I would also advocate for is that you start to get engaged with the growing organ-on-a-chip community. A few weeks ago, a couple of months ago, we launched our Moxie, which is our social media network for organ-on-a-chip technology. Here you can get the latest and greatest on organ chip developments. It's not just for Emulate organ chips, but homebrew systems or other types of organ-on-a-chip technologies that exist. And it's really a great way to connect with other researchers in the field. And it's separated by research or application area, so you get to understand or identify labs out there that are interested in your area of research, have already started or have some experience with organ-on-a-chip technology, and you can also interact with Emulate personnel as well. We're quite active in the community as well, so it's a great forum, and you can sign up for free at moxiecentral.com. So that's the end of my presentation today, and I think what I would do is I will close up with a challenge to the research in vitro cell biology industry. Is that what insights will you uncover with organ chip technology? Now, I've showed you examples of how the technology can be used to extend the functionality, open up new mechanisms, and become a more predictable system for predicting human responses. But again, what we've shown you today is the tip of the iceberg of the potential, and really the organ-on-a-chip technology is something that is only limited by your imagination and ingenuity, and how you can build your own model. So, and really it is up to our research community and our pharma researchers to see how you can push that biology, again, to close that preclinical gap that we can see today. So, and with that, that's all from me.
Thank you, Luke. Great presentation. We will now move to the Q&A portion. And as a reminder, please submit any questions that you have within the Q&A box. Looks like we already have a good start, so I'm going to dive right in. So let's see. So Luke, first question is can we control the frequency and the type of stretch of the chips?
Yes, you can. The Zoe culture modules enables you to be able to control that stretch and the amount of stretch as well. So frequency as well as the intensity of that stretch. Yep.
Great. There are a variety of organ chip companies. What makes Emulate different?
Yeah, good question. I think, listen, what is encouraging is that there are a number of different ways people are bringing these solutions to market, and or trying to expand in vitro cell biology. I would say what makes Emulate different, though, is that we have an all-in-one system. So we the chips, we have the consumables, we have the equipment, but we also have those protocols and that biology as well. So to get customers started really easily and really quickly.
Yes, and our expert team supporting
Exactly.
getting up and running. And
Your team, Melissa, are wonderful.
that's right.
And the other thing I would say is that when we validate our organ models, we really do look at using reference compounds to show how the system is predicting the responses that they expect to see in vivo. And again, the study I showed you with the liver quad culture kit was one example, but all of our organ models have been perturbed with a clinically relevant drug of some sort and shown to replicate that response.
Yes, great. Media question, is there specific media provided by yourself that can be used in the system, or can we use our own media?
Yeah, really good question. In terms of media, so our brain chip actually has, as part of that bio kit, we do provide the media for that particular model. But for the other models, we either point customers to the vendor where you can buy the media, or how to formulate the media yourself.
Great. Okay, volume of effluent. What can be how much volume can be collected?
Good question. So I believe that you could put 4 ml, 4 milliliters per reservoir. So that's kind of the inlet volume, and you're able to collect that on the outlet as well. And again, you can sample over time as you start to collect that effluent. So you can see temporal changes in things that the biology is putting out.
Right. Okay, so question around genetics playing a role in the in vivo responses. And so are multiple cell types collected from one individual, i.e. cells are sharing the same genetic signature?
Yeah, good question. So I would say that in the models that we have built, and actually many of our customers build, they're not often isogenic. So our models, they're primary, and so we have primary hepatocytes from one donor, endothelial cells from another donor. Now that's just how we've built our models. Again, they work, but really what our vision, as well as what our researchers want to do, is drive to isogenic models, so single patient derived models. So how do we match those the hepatocytes to the right endothelial cells, have the right Cooper cells as part of that liver chip that's matched the hepatocytes as well. So that's the goal. Customers have done this. So an example is a publication by Cedars-Sinai where they looked at deriving 2 model types. It was a colon model and a brain chip model, and they did that from iPS cells. So they generated organoids from the iPS cells. So these were truly isogenic models. So it's possible. You just need that expertise to be able to do that, and that's something that Emulate is looking at moving towards.
Okay, great. How many cell types can I seed in each channel?
Oh, so I gave an example of one of our more complex models. That was the brain chip. So and in that we seed we see multiple cell types in the upper chamber. So I would say that it is organ dependent. What we shoot for is looking at the in vivo relevant ratio of cells between different cell types with the functional organ unit that we're trying to model. So that's how we've developed it. But I think there certainly is going to be a limit to the amount of cells and crowding of the cells. But at the moment, as I said, we've seen 2 to 3 cell types, even 4 cell types in the top channel, and that seemed to be fine.
Yes. A follow-up to the isogenic model, is it possible to compare between healthy versus patient?
Oh, yes, absolutely. So, if you're moving to an isogenic model, and you're taking somatic cells and reprogramming them, you can take those cells from a diseased patient. So in the example I shared before, I think it was a Cedars-Sinai publication, they took healthy as well as Parkinson's patients and then generated the models that way. So that's what you can do. When it comes to organoid-derived models, so again, our duodenum and our colon chips are primary biopsied organoids. They're healthy, but we have customers that are also biopsying from individuals that have disease and varying types of disease, and and then modeling that biology on chips. So there's different ways of getting to that question.
Okay. So how are drugs or viruses loaded onto the chips?
Yep. Good question. So we have, again, guidelines for doing this, but essentially your virus, if it's a virus that you're looking at, looking at sort of an AAV infectivity model, which can be done on our liver chip and other organ chips. You basically add your compound or your virus directly to the input reservoir of the chip. So you can dose in different modes. You can do top channel, you can do bottom channel, you can do both channels at the same time. It just depends on where the target is and how you want to design your experiment. But really, that's just done by diluting in the inlet media and then flowing through over time. And again, we have protocols and guidance on how to adapt that, even from a static system to a flow system. There's some complexity there, so we can guide customers.
Great. And a follow-up question here is on flow rates. So what flow rates can be run on the system?
Yep. So we enable from 10 microliters up to 1,000 microliters per hour flowing through each channel. So that's the sort of the boundaries that we've used in our system. And again, for the models and the biology that we're looking at, that's sufficient. In fact, the example I shared previously, the immune cell recruitment at high flow rates provides a sufficient level of shear that drives that selectivity of recruitment. So we're able to see that in our system. But of course, with iterations of the technology and development, we're looking to push that further down the road.
Yes. Okay. We have time for about one more question. Let me go through our list here. All right. So where do you get your cells from?
Oh, good question. So Emulate, we have a number of qualified vendors that we work with, and we select these vendors, based on their capabilities to isolate certain types of primary cells or their ability to differentiate any sort of certain types of iPSC cells. What I will say is that it is very important, the biology itself. Often, when it comes to things like hepatocytes or Cooper cells, not all vendors are alike, and you're able to see differences in our system. The system is quite sensitive at looking at that biology. So, for our biokits and our pre-boxed organ models, we have a number of different vendors that we use, and we pre-qualify those cells and we bring it into Emulate as part of the product. So that's our mechanism. But as I mentioned, there's, if you have a preferred cell vendor and you already have a model established on a static system, the next logical thing is just to use that same material and apply it to chip.
Okay. Thank you. Thanks for the discussion, Luke. And at this time, we'd like to thank everyone for your attendance and fantastic questions. As mentioned at the start, the webinar is recorded and it will be sent via email after the webinar concludes. Thank you.
Thanks, Melissa. Thank you, everyone.